was looking over the slide set and thinking back to what we were covering before the exam. So this is like a whole week ago, right? We were looking at this idea of how the plants and different fungi, one that's more cooperative with sharing, one's less cooperative. And I can't remember, and I didn't want to go through my recording. I can't remember how clearly I was trying to make a certain point, and I'm not sure I made it clearly enough. So I want to just revisit that for a second before we move into the new stuff. And so this is the idea of this was with phosphorus. So, how much phosphorus would the mycorrhizal fungi give to the plant in this study? And they did a split root design, which is being shown up in the corner there. So we talked about that last Monday, and that on one side they would have a more cooperative fungus in the soil, and then on the other, a less cooperative. And they were deciding that idea based on phosphorus, like how well was the fungus giving up the phosphorus, and how much carbon did the plant have to give back in return? And those were the deciding factors on, more versus less cooperative. And what they were finding is that the less cooperative fungus means that, it was like greedy for the carbon um, when it was by itself, but when they had it in the split root design where they had the less and more both there, they were showing that the less cooperative was starting to become more generous in the sense that the plant didn't have to pay as much in carbon, I guess is a way to put that. And so this idea that when you have a mixture of fungi, that you can get this kind of checks and balances. And if you kind of think of it as market economy, I think this next slide, where is it? Yeah. So the addition of a second mycorrhizal fungus likely increased the overall supply of phosphorus. So there's more phosphorus for the plant to get access to, and that drove the price of phosphorus down, which means the plant didn't have to pay as much in carbon. And that's the point. So the cost in carbon was reduced due to a more competitive market, two fungi instead of just that one. And so I wasn't sure how clearly I made that point. And then the last point, they're saying this could be really important for when you think about more natural ecosystems, that, you get this diversity of mycorrhizal fungi. And that's kind of a good thing because it makes sure that you don't have really uncooperative fungi that are taking advantage of the plant. So you won't get any one partner in this relationship constantly winning and constantly losing. You have this kind of checks and balances. Okay. So I wanted to just revisit that. And then I think we left off looking at this idea. So we were looking at, we've been covering mycorrhizal fungi and common mycorrhizal networks. And then this study was a humongous study that was done forests worldwide by a lot of different scientists, as you can see here. It's amazing. And we were getting into this idea that when you look at forests that are in this area, this latitude, we have, and above and more north of us, we get a fair dose of ectomycorrhizal fungi. In the temperate and boreal, but you get more arbuscular mycorrhizae in tropical and subtropical, and then you kind of get mixtures. There are definitely forests that have both. And so they are starting to come to the conclusion that, when you have a mixture of these types of fungi, that they often can be more productive than if you just have one or the other. And then I think we left off on this slide where we were looking at this idea of, how come some forests are more diverse in terms of plant species that you can see? Why are they more diverse versus other forest systems? One of the driving hypotheses is that you have pathogens that are definitely influencing what plants survive and how many survive. And so this idea of pathogens tend to, be specific to certain plant species, like that pine beetle infestation, it has certain species that it favors. And so if you have kin in, the same lodgepole pine trees all together, it's really easy for the pathogen to get into that clump of lodgepole pine and cause a lot of disease and death. But if you have diversity of plant species, it's harder for the pathogen to really do substantial damage just because there's so much diversity. So this became known as the negative conspecific density dependence. Conspecific just means the same species, so a bunch of lodgepole pines, the mother tree and maybe a bunch of seedlings, that you just generally don't get that if there's a lot of pathogen pressure. Then so that's been known for a while. That hypothesis is pretty out there and accepted. But what they've been noticing is that you get this CDD really stronger when you get into like the equatorial belt. If you think about if you've ever had the ability or the good luck, good fortune to get into a tropical rainforest, for example, where there's just such incredible diversity of plants. And they say that it's because the negative CDD is stronger at those lower latitudes and in wetter, warmer sites, which means you end up with more plant diversity. And that helps explain why they do see that in the tropical areas. You get incredible plant diversity. And that's pretty well accepted. But now they're saying, okay, what role does the mycorrhizal fungi play in all of this? Does it play a role? And so they've been studying this recently and looking at it's kind of the opposite force now, where you can have the mycorrhizal fungi. You can imagine those lodgepole pine trees, for example. They share and have like favorite mycorrhizal fungi that they hang out with. And so if you have a lot of those mycorrhizal fungi, you could see where lodgepole pines might start hanging out together because they share that really nice mycorrhizal network. And so if the mycorrhizal fungi are helping to increase the same species, conspecifics, if they're helping all those lodgepole pine seedlings to hang out there, then they might have an impact on that negative CBD idea. And so again, it's kind of like, the bad versus the good. You got the pathogens coming in and their selection pressure of wiping out conspecifics, and then they're saying, but the mycorrhizal fungi actually support conspecifics. And so you have this yin and yang type idea, I guess. And so in this particular paper, which I'm not going to assign, I'm just going to summarize what they were finding, is that you get differences depending on the type of fungi that are present. They show the arbuscular versus the ectomycorrhizal tree species differ in the strength. Looking at the saplings, the conspecific, seedlings in that area. And so they say, we think, the major difference there is that the mycorrhizal types are really influencing what's going on. And that really hasn't been something that's been looked at in detail when they came up with the Janssen CDD hypothesis. They really hadn't factored in anything else. And so this paper, they're saying, we need to think about the mycorrhizal types, and they may be part of this whole idea of the global forest ecosystems, why we see these different types of ecosystems. So variation in forest diversity and ultimately the latitudinal gradient that they see in the tree species diversity as they study all these different forest systems. And so they're saying, hey, we need to pay attention to these mycorrhizal fungi. And, they say we have to think about other soil microbes too, but they were mainly focusing on the mycorrhizal fungi in this idea. And that they really could be driving a lot of the selection pressure and what we see in these different geographic locations. So I was super excited when this paper came out and I found it because that has been something that has been hinted at quite a bit that we have to pay attention to the mycorrhizal fungi in helping to explain what we see, what's going on above ground. And it's very complicated. I'm not saying, there's so much more that they have to study on all of this, but you know, I think you can appreciate with what you've already learned about common mycorrhizal networks that it certainly would have the ability to provide a strong selection pressure on these different forest ecosystems. So hopefully this will get more attention as ecologists are starting to look at in more detail at forest systems, that they'll start to pay attention a little bit more to what's happening underground, especially with the common mycorrhizal network. Okay. So that's where we left off, thinking about that idea. Um, I guess I kind of want to switch gears a little bit, still looking at, this slide set has a lot to do with kin recognition. Wanted to also get into niche partitioning too, and how that might play a role. And so with kin recognition, this paper was published in 2018. So they're saying, okay, it seems kin recognition is a thing, at least with many plants that have been studied. It looks like certain species are definitely able to recognize their own species and others, non-kin. But now they're thinking, okay, how do we take this information? Can we apply it to improve food production? So looking at the agricultural production. And so these two papers looking at, how should we think about this? Would it be better to plant kin all next to each other? If you think about how a lot of agriculture is currently done with big monoculture fields, right? Acres and acres of just the same, corn or soybean. And is it better to have closely related corn plants together? Maybe not related. And this kind of is getting at kin recognition, but also looking at niche partitioning. So we have to kind of think about, and competition, we have to think about all of these ideas. So now I'm excited to see that they're starting to think about, okay, we're teasing out this information. It looks like there's kin recognition going on and non-kin. So, okay, let's take it and apply it and try to help improve food production. So one of the first studies was that rice study that was coming out with what happens if you plant rice plants that are, siblings with each other versus non-siblings. And this is just a figure showing what kind of a summary of what they found. And so here, when they were growing rice that was the same, siblings, what they're trying to show with this picture is that they got really good yield of grain. Okay. And then this idea down here in the root zone, they're saying that this particular chemical called allantoin was not nearly as high as if you had two unrelated rice plants growing right next to each other. When you had that situation, you had allantoin levels being excreted by the roots that went up. And they noticed that. And then they also noticed a big difference in overall yield, in that they were not getting the yield in rice if they were non-kin planted next to each other. So based on this study, they were saying that if you have kin right next to each other in that monoculture, you ended up with less root competition. So if you think about rice, it's kind of plant specific. It's like, okay, for rice, we want the grains above ground. And so we don't want to really do anything with the roots. So we want to make sure that we get the rice. And you don't want to have a lot of stuff going on with the roots. And so they're saying you had less root competition when they were kin. And they noticed this allantoin chemical in particular was definitely different between if they were with kin versus non-kin. And so they're still exploring that. Like, what does that mean? They're expecting or hypothesizing that that increase in allantoin was a signal to the plant to, compete and really start to, fight each other for nutrients. But they admitted they still have to do some work on that a little bit to figure out what that's all about. So this is kind of their summary of that paper, and that, they're saying this could be super important for agriculture to understand the kin versus non-kin and thinking about what plants you're growing, how you want to grow them. And, it's monoculture growing in many systems. That's what they do. They just have one plant and they tend to pack them in very closely to each other. So they're in close proximity. They can't help but interact with each other. They're forced into it by how we plant them. And so they're expecting that that allantoin is acting as a chemical cue. So they say, we really do have to think about how are we going to plant our fields? What are we going for? And just be a lot more thoughtful about that when you're growing plants in big monoculture settings. So this figure is taken from a different paper that was published just a couple years ago. And they reference that paper we just talked about in figure A here, where they, talk about this idea of um, the non-kin interaction with that increased allantoin, kind of triggering that competitive interaction, especially with the roots. And then some of the other figures here we're going to get into a little bit more in subsequent slides here. So in figure B, they're looking at kin, and they're trying to show by those red dotted lines, they're trying to show that when you have kin jammed next to each other like that, like in a monoculture egg, that they can actually reorient their leaves so they're not shading each other. And so they wanted to study that a little bit more. We're going to get into that a little bit. And then another concept they've been looking at is that, so much of our food production is we want the plants to flower and then get pollinated so that the fruit can get made so we can harvest that and eat it. And so here in this figure C down here, they're showing that if you have kin growing together, so over here, what they're trying to show in this graphic is that they've actually seen the flowers be more showy if it's kin next to kin. Like the flowers, they invest more in flower display. Look at our big, beautiful flowers. And when you have a lot of plants all close together, all producing these beautiful flowers, it's very attractive. Pollinators are often very visual, and so they'll pick up on that. And so they're saying if you get more floral rewards if you have kin next to kin when it comes time to flower, because they're really advertising really well for the pollinator to come in, and they're not competing, they're putting less resources in competition. They're cooperating with the flower display. Whereas if you have two unrelated, they're not siblings, they're trying to show that you wouldn't get that increased floral display, you'd probably get more competition. So this is something that they're investigating, and some of the papers that have come out are supporting the idea that kin will do this. They will, kin to kin, they will ramp up their flower display because even, it really does, it pays to advertise. If you get that nice big floral display, you're going to get nice pollination. And for them, the goal of the plant is to get their seeds or to get their genes into the next generation. So you need to get pollinated, you need to form the fruit where the seeds will be located. So they're looking at these ideas, and we're going to cover some of these. And so this one is looking at what I just, figure C with this floral traction. And so kin discrimination allows plants to modify investment towards pollinator attraction. So this was published back in 2018. And their study, it paid off with, their hypothesis, the data supported it. So as predicted, individuals growing with kin, particularly if they were at high density, packed together like in a monoculture, they produced larger floral displays than those growing with non-kin, exemplifying the importance of kin recognition in the evolution of plant reproductive strategies. So this paper was not focused necessarily on agriculture, but they were saying, this idea of growing closely together is the idea of when you grow plants for food production in farming, you definitely have that situation. And then this paper came along around about the same time. So if you get pollinators coming along, hopefully pollination will occur. Then you get fruit and seed formation. And then they're saying seeds integrate biological information about conspecific and other specific allospecific neighbors. So they're even finding seeds are able to pick up on this. Seeds which are embryonic stage of plants need to make decisions about the timing of emergence in response to environmental cues. Should I go ahead and germinate or not? And so they were looking at the timing of emergence. And they were saying if you, grew next to some non-kin, trifolium, so Plantago versus Trifolium, those are two different genera. And then they were looking at relatedness of the Plantago, if you had kin versus non-kin. So where I'm losing my eyes are jumping. So the Plantago seed and the presence of seeds of Trifolium did not on their own influence the timing of emergence. However, when encountering a Trifolium seed, a Plantago seed emerged faster in the presence of a sibling than in the presence of a non-sibling. Then they took some water extracts from the seeds and tested just the extracts, and they saw the same result. So what they're saying is that the Plantago seeds were actually using information about who's around them in the seed stage. Okay, so this was just seeds getting planted and watered. So they were becoming active. They weren't dormant. They were starting to activate and germinate. And they're saying, yeah, the Plantago seeds, they were figuring out who was around them and if they were related or not. And so this idea that even that young, once, the seeds are activated at that very initial stage, they're able to tell kin versus non-kin. And so this paper was kind of blew a lot of people away because that was not really thought to be happening so early in the stage of life. Okay, so let's think a little bit. We have to kind of layer another idea. So we've been talking about self-recognition, kin recognition, non-kin recognition, mutual benefit hypothesis. Let's layer something else now, niche partitioning. And this is, let's compare the two. So kin selection, we've been talking quite a bit about that. And, this idea that it makes sense that they can recognize and cooperate with their kin helps to get your genes into the next generation. Even if it's not your own, at least your species is getting into the next generation with kin selection. Niche partitioning. This is the idea of reducing competition by using different resources. So if you're next to each other, you're in a cluster of plants, you reduce your competition by, like, well, I'll take this patch of soil, you take that patch of soil. You're niche partitioning. Your roots are like in two different patches. You're doing a niche partitioning thing. So that would be a good example. Or I'll grow my leaves this way, you grow your leaves that way. And that would be a different light resource. Okay. So this idea, niche partitioning is you use different resources to reduce competition, essentially, is what you're going for there. And they say if plants can do that, in a patch of soil, if they're all together, if they can do this niche partitioning, it should help them share the environment more efficiently. They're not all fighting for the same resources. You can see this happening with same species plants or diverse mixture. Okay, so that you can apply this concept really to any mixture. So with kin selection and niche partitioning, they've been, as we've been looking so far, looking at root structure is one way to kind of get at this idea. And, looking at their root structures, are they going to be going for the same resources? Do they have the ability to maybe do some niche partitioning? And so these visual images are just trying to show different root structures, different morphologies, different depths. How big do they get? How long do they get? And so they've been studying root systems for niche partitioning. That's quite a bit of what they're doing, but that's not the only thing. But we'll look at that idea. Okay, so that, I think if you think about it for a sec, that kind of makes sense that the root systems, you definitely could see niche partitioning. I think I showed this slide already. This is getting at the idea of if you have kin on the left, you should see, if it's kin selection, you should see less competition in the roots. So that's what they're trying to show here. Less competition. They're politely restraining their growth. They're being polite. Whereas if they're non-kin, you see a more aggressive root growth. And then they say, okay, well, then let's think about niche partitioning. So figure B is, okay, let's look at these plants and think about niche partitioning. If they have the same root system, if they're kin, right, they're going to have the same root system, at least initially. They're going to grow very similar to each other because they're siblings. You would predict there would be stronger competition then because they're same root system, same depth, they grow the same morphology. They're probably going to compete with each other. And if you had different species with slightly different root systems, then they could do niche partitioning. And that would be a good thing. It would be more of a cooperative interaction because they're like, well, I'll use this patch and well, I'm going to use this patch. And so you would say they're facilitating each other, everything's good. And so these ideas kind of are opposites in terms of what you would predict, depending on if you're thinking about kin selection theory or niche partitioning. And so there's been a lot of debate about this in the plant world. And they are, Susan Dudley was writing a paper about this idea and, saying, well, it's very dependent on the environment, how these will play out. And, saying which one's more important, it really depends on the environment, what's going on in that ecosystem. And she's proposing it's really not that simple. Of course, everything in science and nature is never simple. She's saying, maybe we should say that these two ideas are coexisting on, a day-to-day basis. You can have kind of a continuum of things happening, and they can jointly influence what's going on in that ecosystem. And so she's raising the point that, we tend to simplify things and like just focus on kin selection. Only that's happening. And then other people might say, well, it's niche partitioning. That's what's happening. And so we tend to kind of simplify things down. And she's saying we probably can't be that simple in how we look at these systems. The either or nature is probably too simplified. And so this was a kind of like a review paper where she was looking at this. And then some other scientists just recently came up and said, yeah, that's true. We need to really think about the ecological context and kind of tease apart what's really happening here. So I'll show you a visual here. And this actually includes a third thing, facilitation. So you have niche partitioning, facilitation, and kin selection. So the niche partitioning, I don't know if you can tell by their drawings what plants those are, but it's corn and then bean and then squash. So that's the three sisters idea, companion planting with the three sisters. And they're saying that is a really good example of niche partitioning, in their opinion, because you have different things going on. You have the corn, their root system, all the root systems are different. They're unrelated to each other. So, and they have different root systems. Look at the difference in height and their growth pattern. They don't really do a good job with the squash, but you have to imagine the squash leaves are kind of sprawling along the ground. Okay. So they didn't really, they should have put that in there, but they didn't. Oh, I guess they did in facilitation. Well, you can see the squash there. And so they're saying, if you have these different, you have functional diversity in the way the plants grow and how they're going to use the resources there. And they're saying if you grow these three together, you have increased, like the beans are busy doing, the microbes with the beans, the bacteria are doing nitrogen fixation. So you have nitrogen being added into the soil. Well, corn is very hungry for nitrogen. They need a lot of nitrogen to complete their life cycle. So that's great for the corn. And then the squash can't, I mean, I'm sorry, they should have put the bean next to the corn because the beans start to grow and wind around the corn. They use it as a support. So the drawing's not the best. It's the best one I could come up with, and I can't draw, so I didn't do it myself. But anyways, the beans should be crawling up the corn so that they can go more upright, get to the sun, and they're doing biological nitrogen fixation, which the corn really needs all that nitrogen. And then the squash, because they grow more horizontal onto the ground, they're keeping the soil shaded, cool, moist, more easy access to water. And they're using different spatial areas above ground, and so there's less competition for light. So you can see that they're doing this process of niche partitioning. Facilitation is kind of like because of their activities, they're actually helping each other. That's kind of the concept with facilitation. Just by their particular activities, the output from that can help other nearby species. And so that's what they're getting at with facilitation. So here they're focusing on more of the ability of the plants to bring up certain minerals and water from the soil. You have symbiosis. They can share nitrogen and water. Okay, so just by their functional activities, they're kind of helping each other in that regard. And then kin selection, they're saying, you can have, when they're kin, they should not compete. They're more polite with each other versus if they're non-kin. And so this is what a lot of scientists are looking at when they look at ag systems. Like, what would be best? Should we do companion planting in a thoughtful way, pick the right companions? Should we do monocultures? Should we pick plants that facilitate each other? So one of the earlier studies on this idea was this one. This was 2017. So light mediated self-organization of sunflower stands increases oil yield. So this is when you grow sunflowers in ag, you're going for the seeds or the oil that you'll get from the seeds when you harvest the seeds. And I don't know if you can tell in that picture. Let me see if I can blow it up a little bit. So when they plant them really densely close to each other, I don't know if you can tell, but in this experiment in figure A, what they did is they planted them really tightly together compared to not so tightly together. And then the vertical forced means they put wire, like cages, around the sunflower plant so they couldn't move. They could just grow vertical, but that's it. You're just growing vertical. So that's what that means. And then this field we'll plant you to the same density, but we'll leave you alone. And let's just see what you do. And I don't know if you can tell, but the stems are inclined, kind of they alternate down the row. And it was really it was surprising the results and the conclusions they were drawing because this was never done before. They thought if you plant at such high density, your yield is going to crash. A lot of people were like, yeah, that's not going to work. You're going to have far less oil production, yield, I should say. And so here they have some nice data, oil yield and stand density, 10 plants per square meter or 14 plants per square meter. And you can clearly see the difference. You could see that they were doing this, if they had the incline, so the clear bars in both cases, either at the 10 or the 14, but it really shot up in the higher density. You saw that they, did that inclination growth pattern where they were alternating which way they were leaning down the row. And then that allowed those plants to produce more oil. So they were more efficient and vigorous, even at really high density of plants per square meter. So this was a little surprising because it was not the conventional way to grow sunflower in that dense of a planting of the plants. So let's look at this too. So this is another image. Maybe you can see it a little bit better here, how the ones on the left are vertical, and then the ones on the right, you can kind of see the tilting pattern. And they're trying to show it with the red-green bars there, the difference in the tilting pattern. They're leaning. And then here, it's kind of fun. They're saying, which way are they inclined? Inclined to the north, it's red, green is inclined to the south, and gray is not inclined. And they're following it. Well, there's two options here. You have the density of the plants. So they had three different densities of how many plants per square meter. And you can see higher density of the plants, the more they did the tilting back, north, south, north, south. And then, initially, when they first germinated and grew, 27 days after emergence from the seed state, or from the soil, I should say, when they emerged from the soil, you can see they were kind of doing that. And then as they grew up, they really got into it and started to do the leading north, south, alternating like they're trying to show in figure A there. So they just naturally went into this pattern of leaning in like that, and it was fine because it resulted in the benefit of greater oil production, which of course for egg, that's what you're going for. So then they were like, okay, it's great we're getting this outcome, that means we can grow more sunflower per square meter, get more oil out of that. But why? I mean, how are the plants doing that? And this is where we come back to the idea of phytochrome and looking at the quality of light these plants are exposed to when they're jammed next to each other like that. They tried different quality of light. They were trying to find out like what's the plant paying attention to do this growth pattern. They tried blue light. So you can see blue light really didn't do anything. Whoops, sorry. So blue light didn't have them do any inclination of growth. They didn't lean. Here they show what happens if we simulate shading of the leaves. Is that going to trigger them to lean? Yes. You definitely see a big, significant increase in their leaning away from the neighbor. And then they said, okay, well, leaf shading, the phytochromes are really picking up on that. Am I in sun? Am I in shade? It's the phytochromes. So then they started to look at that. So now figure C, now we're thinking about red, far red ratios. Okay, so let's look at that. If you have low ratio, so this first bar, less than 0.1, what that means is that you don't have much red light, okay? So if you don't have much red light, thinking about the phytochromes, that means you're going to have more PR. Right? So your red far red ratio on the left bar is saying there wasn't much red light. So that's basically saying, well, I'm getting shaded, so I'm going to incline. And then as you go to the next two bars on that graph, they're getting more red light. And so they're not inclining as much. So basically, what the sunflowers were doing is like they're using their phytochromes and they're like, oh, I'm getting shaded. I'll lean. Okay, now it's good. And then they would grow and get good oil production. But if they were sensing with their phytochromes, I'm in light, then they didn't really do much. So the trigger was if they think they're in the shade, then they started to bend away from upright. That was what was going on there. And so it was a positive feedback with phytochrome in the mix there, helping the plant decide, should I just keep growing up because I'm in the sun, everything's good, or am I getting shaded out so I should lean? And then I'll get to some sun. Okay, so that's what was going on there. And so this is the full abstract there. You can read that too when you're studying. So this was kind of a interesting result that was really surprising. A lot of scientists that are in ag sciences didn't think that this would be such a difference. I mean, look at that, 19 and 47% more oil if they leaned compared to if they stayed upright. So let them lean. So that gets at the idea of, okay, it looks like there's photoreceptors involved in helping plants figure out kin versus non-kin. So we've already looked at how plants can figure out kin. You remember with the sagebrush experiments, what was the chemical cue? What was going on there? Yeah, right. So the volatiles, right? The volatiles that would be passed to the neighbors and that would help them defend themselves. So it was a volatile signal for kin, like, they use the same chemotypes. And then Dudley's work with the root systems, excreting chemicals and the root systems could pick up on that and go, oh, you're kin, and then they'd politely restrain their growth. Now they're getting at, okay, what else could be involved in kin recognition? And so they started looking at phytochromes because that's a big one with plants. Phytochromes are super important for plants. And so they said, well, let's look at that. And then they threw in cryptochromes and phototropins, which are the ones that can detect blue light. And so we already saw with the sunflower, the plant really wasn't using was it the phototropin? I'm blanking on which blue light. Yeah, I can't remember which one. Sorry. It's one of those. Anyways, here. So here they were looking at, they didn't use a like standard crop plant for this. They went with Arabidopsis, which is the guinea pig plant. But, they're easy to grow. They're really tiny plants, so it's easy to do these experiments pretty cheaply. And so they used Arabidopsis instead of a true crop plant. And then they started looking at, let's plant them really close together, like in an ag field. And let's play with if they're next to kin or non-kin and see what happens. And so they had some mutants thrown in to get at some of the, conclusions. Let's see what we can do with this. And so they were finding that if they were kin, non-mutant kin, they were able to actually use their phytochromes to figure out where they are in space next to their neighbor. And if they were kin, they were actually using that information to grow into separate three-dimensional spaces so they each had access to good light. So they were not shading each other. Okay. So leaf position responses, that's what they were looking at. So light signals from neighbors are known to shape a more competitive plant body. Here we show that photosensory receptors mediate cooperative rather than competitive interactions among kin. And so they were looking at how are plants, when you look at the above ground portion of the plant, besides the sagebrush idea with all the volatiles going on, where volatiles are being exchanged, is there other ways of the plant being able to sense what's going on around them? And then you have to throw in this idea of, are they growing next to kin or non-kin? How does the light shining on them and their use of that light vary? So with that, it's 10:02. Why don't we take a break and come back at 3 and we'll see how much further I can go. So let's take a little break. All right. So just wanted to go back to this one just so to clarify this idea here that we were talking about with the sunflower tilting like that, that's like niche partitioning, right? They were, accessing different pools of light. So I'm trying to make the point that they were busy doing that and it was very productive for oil production. All right, let's go back to where were we? Okay, Arabidopsis. So here, so we're still thinking about phytochrome and the ratios of those pigments and the light. So let's, let's think about the light striking the plant body. So if you have a bunch of Arabidopsis plants jammed next to each other and they're busy living their life, they're going to be doing photosynthesis. And plants that do photosynthesis, they love red and blue light. So if they're absorbing red light, any leaves that are getting shaded below that are going to get less of that red light because the upper leaves took it and used most of it. And so less gets passed through, but they'll let far red light go through. And so plants are paying attention to that as a way to tell, am I in light? Am I in dark? What's going on? They're using their phytochrome pigments. So they were looking at that. And then they were also wondering about blue light. Is blue light going to be influencing what's happening here when you jam all of these Arabidopsis next to each other? And what they were finding now in the sunflower, when they did the blue light didn't have any impact on that study. But here they were showing that they did see blue light playing a role. So let me finish the thought here. So they were measuring redirection of growth. And so they know that plants, when they're trying to avoid shade, like if they're a plant that needs a fair dose of sun and they don't want to be always in the shade, there's this mechanism called shade avoidance that plants will do. And they're using their phytochrome and their blue light photoreceptors and measuring what's going on with far red light, what's going on with blue light, and they can use that information to avoid the shade if they need to, if they're that type of plant. So what they did in this study, basically the big picture idea was they had pots of Arabidopsis. So each plant got their own pot, but they jammed all the pots really close to each other in a line. And they did it in doses of 7 plants per treatment. Okay. So if they're in pots, that means their roots were not able to interact with each other. So we are not talking about underground chemical signals. They're really focusing in on what's going on with light resources and how are the plants responding to that if they're kin versus non-kin. All right. So here you can see a little bit of the experimental setup. So what they did, I don't know if you can clearly see, but there's several, looks like this figure B, day 5, I can see 3 pots right next to each other. So they're jammed next to each other, and you can see how Arabidopsis kind of has that spreading, a little bit of a spreading growth pattern with its petiole and leaves. So that red leaf, the red colored leaf, if you're not red green colorblind, the red colored leaf, they're trying to show how it shifted its position from day 5 to day 7. I don't know. I can't reach that far. Um, you can see the dotted light. Dotted here is where they said that's where the leaf was at day 5. And then at day 7, it had shifted. Is that visible to your eyes where you're sitting? Okay. So they're trying to show you how the leaves could shift in response to detecting, neighbors and the different qualities of light. So the black circles here on this graph in figure A, they're looking at days of growth on the x-axis, and then the leaf position ratio. So basically, I define the leaf position ratio here in figure 1. It says leaf position ratio. Whoops, didn't mean to do that. The number of leaves growing away from the neighbors compared to the number of leaves toward the neighbors. That's how they defined leaf position ratios. So the higher the leaf position ratio means that the leaves were growing away. They're growing away from the neighbors. They're trying to get away. And so you can see compared to controls, all the black circles, you can see how much higher that was. Oops. In figure, boy, doesn't want to pay attention. Oh my goodness, really? There we go. Um, with kin. So with kin, they were really moving their leaves away from their neighbors. And that's what's being shown in figure 2A. And then figure 1A is also showing that in a bar graph form, and where you have the kin really significantly moving their leaves away from their neighbors compared to if they were non-kin jammed together in those 7 pots in a row. Figure 2C, that's how they did their measurements. They actually had like a transparent thing that had an X pattern on it, that red X pattern, and they'd actually look down on the Arabidopsis plant, and that's how they would assess which leaves were growing toward kin versus away from kin. So this is some of the data that they were collecting here. That's how they were doing it. And I think you can see here, as you look, so towards the neighbor is kind of this left right. If you see more leaves in those zones, they're growing towards, and then if kind of the north south is if they're growing away. They're also trying to show here that they were paying attention. It's really kind of hard to see. But they have some arrows here, the red arrows, they're pointing at the petioles and then the leaf. So basically, most Arabidopsis leaves grow horizontal, but they were showing sometimes they would torque their petiole and leaf, so they'd be more vertical instead of horizontal. So they were not only just kind of growing away from a neighbor, but they could twist and so that they weren't shading their neighbor in that way. So they were really taking some nice detailed data or measurements for their data collection. So by the way here, single accession, they say that means kin. And then multiple accession, that means there's a bunch of different non-related, non-siblings. I thought this was really interesting. They were also looking at age of the plants growing next to each other. Like what happens if we offset when we get them germinating? So some plants are a little older than some of the others in that line of plants. And so the fancy word for that is, think you say cotaneous. Does anyone know? I don't know. I'll go with cotaneous. That means they were at the same height. They were the same age and at the same height. And you can see if they were, then they really made an effort, so to speak, to grow away from their neighbors. But if they were all different sizes and different ages, they couldn't recognize each other. They just in this study, they just like, oh, well, they couldn't figure it out. So they're saying it's kind of important that when you're planting seeds and if you want this impact, where they're not going to shade each other, you make sure you're planting all the seeds at the same time and that you get, the same age growth happening all at once. It's all the same. And then that way you will still get the shade. They won't be shading their neighbors. Does that make sense? Okay, I thought that was really interesting that they thought of that. And so, okay, so it looks like they're being really good about not shading their neighbors if they are kin. Then they said, they were testing a mutant. So the SAV3 mutant is a mutant that doesn't produce auxin, which means they wouldn't be able to really do that growth movement away. Like they're stuck in that regard. So the wild type was normal, and then the SAV3 mutant is an auxin synthesis gene. Okay. And so again, they were just saying, a little bit more detail of what's really going on here. And they're finding out that, the ability to produce auxin to full strength is important. And then they were looking at some of this data is also showing that if, so if you're thinking about it, on Arabidopsis plants, like, oh, I'm going to move my leaf so my kin neighbor isn't getting shaded, but that might shade one of your leaves, right? So, especially as a plant gets older, you'll have younger leaves on top of older leaves. And if they're trying to avoid their neighbor, might they do self-shading? And so they were like, ooh, could that be a bad thing? Like if they self-shade too much, maybe they won't be productive. And so then they were looking at that. With this study, they were finding that it was better not to shade your neighbor. It worked where the decrease in mutual shading outweighed any increase or the increase in self shading. So it didn't really matter that they were self shading. They still were able to show a net benefit if there was that reduction in, don't shade your neighbor if they're kin. So they're like, yeah, shading can be an issue though, because then you don't do as much photosynthesis, of course, so that could be a problem. So, okay, what kind of impact is that going to have all the way through? So they followed the plants in another experiment all the way through to seed production. And so they're measuring seeds, like number of seeds per plant, if they're kin versus a mutant in this case, and trying to figure out, is this going to be a problem if they're being so polite and trying not to shade their neighbors, but then they end up shading themselves? That, could be a problem. When they looked at the data though, they said when you're growing with kin neighbors, seed yield was higher. Even though they were doing some self-shading, it still worked out that you had seed yield that was significantly higher in the wild type compared to the ones that weren't doing that movement away. And so they said, yeah, this is really, they're figuring out that they have kin next to them, and then they're behaving in response to that by not shading their kin. And even the little bit of self shading they were doing, it paid off in the end. They still were able to show that self shading didn't really impact the seed yield compared to the SAV3 mutant that wasn't able to turn and move its leaves. And so they're like, even with self shading, you still got better seed yield. So Really, really well thought out study. Really nice. They did like 8 experiments. I'm not going to assign this paper to read. It was big and they did a ton of experiments. So I'm taking kind of the main ideas from this. Really interesting study. So they came to the conclusions. Okay, they said, yep, we were doing pots of plants. So there wasn't this chemical exchange underground. So we can ignore that for this set of data. Of course, there could have been volatiles, but nothing underground. Oh, one of the experiments that I'm not really going to talk about too much, though, they did another set of experiments where they used plastic filters rather than a neighboring plant, which gets at the idea of could there be a volatile? And they were able to show with the plastic filters, simulating a shading versus non-shading idea going on, that they ruled out volatiles, above ground volatiles. So they ruled out, because they're in pots, any kind of root communication. They ruled out the above ground with these plastic filter tests that they did. And then they said if the plant is showing that, if they're next to kin, they rearrange their leaves, they politely grow, they share different pools of light. So they are doing niche partitioning. They're avoiding that competition. And in the end, they still increase their fitness by doing that. These cooperative interactions would be particularly important in agricultural crops involving genetically similar individuals grown at high densities. And so this, even though it was done with Arabidopsis, it was a really, really nice study to get at some of these ideas of how do we do this? How do we do this for agriculture? And, they're coming to the conclusion that, yeah, doing rows of kin would, yeah, if you plant them all at the same time, according to this data, get them all planted at the same time so they're the same height. We don't know if all plants behave that way, where they can't recognize different heights of their own kin, but anyways, in this study, they gave the suggestion to do that. And so this was one of the first studies where they were looking at light intensities and thinking about that. And then, that really does have an impact, right? Because if you have a bunch of plants all jammed together, they're going to be vying for similar pools of light. And so that's what we do in conventional ag. We jam them all together in these rows. And so this was an important study looking at how does that impact what kind of light they're seeing, and then how will they behave in response to that, if they're kin versus non-kin or mutants. Okay, so really, really nice study. So give that some thought. I'm fading. Are you okay if we finish a little early today? Thank you for your patience on that. I hope to be back to 100% on Wednesday. And we'll continue. There's just a few last things to say in this slide set, and then we'll shift into the next slide set. We also have to start thinking about group paper summaries. So we'll talk a little bit more about that on Wednesday, which means we need to have people here so that they can participate. So thank you all for coming. And we might rearrange who's in what, we might, yeah, not really. Yeah. And I'm gonna, Anthony usually sits at your table, but he's often a no-show. So I'm gonna send him an email saying, hey, it's gonna be group time here. Cause I think the last few weeks of the semester, we're gonna do more group stuff, and it'd be helpful if people participated if they want the points. So final. So as far as more graded stuff, like we're going to do group activities, and then I'm going to be assigning some like very short, like EduCat things for you to respond to, but it'll be like homework stuff. Yeah. So that kind of stuff is coming into play soon.